Good afternoon, everybody. I am Irit Rutman-Halili from Hemdat Academic College. I would like to tell you about my research with my students, Moria Ifrach, Adi Danino, and Moria Sofer. The title of my work is The Effect of Caffeine on Starch Digestion. Caffeine is a natural compound found in a number of plant species, including coffee, tea, and cocoa. It is a central nervous system stimulant of the methylxanthine class and is probably the world's most widely consumed psychoactive drug. However, unlike many other psychoactive substances, it is legal and unregulated in nearly all parts of the world. The ingestion of caffeine in the form of common beverages such as coffee and tea, particularly at higher doses, was previously considered to have the potential for causing harmful effects. For instance, headache, chest pain, restlessness, and heart rate increase. However, recent studies have provided evidence of several health benefits of caffeine. It acts as a central nervous system stimulant that is known for its ability to increase energy levels. Some studies suggest that consuming coffee regularly could be associated with a decreased risk of developing type 2 diabetes over the long term. The mechanism of action in this regard is unclear, but may involve stimulation of insulin-producing pancreatic beta cells. Diabetes is one of the deadliest diseases affecting the body's vital systems, such as kidney, eyes, and heart. Complex carbohydrates with a high glycemic index, for instance, white rice, potato, and pasta from white flour, contain high concentration of starch. These carbohydrates are considered as harmful to diabetics and also have the potential to accelerate the transition of pre-diabetes to diabetes. In the human digestive system, alpha amylase performs the breakdown of carbohydrates. It is found in mouse saliva and in pancreatic juice. Alpha amylase hydrolyzes alpha bonds of large alpha-linked polysaccharides, such as starch and glycogen, and plays an important role in the digestion and metabolism of dietary carbohydrates. Since a carbohydrate-rich diet encourages the development of diabetes, the aim of our study was to find a food ingredient with the potential to inhibit carbohydrate breakdown. In other words, that would inhibit the activity of alpha amylase enzyme. Recent studies have reported that caffeine alters saliva alpha amylase activity. However, the nature of this alteration is still not clear. Some studies reported that caffeine increased saliva alpha amylase activity, while in contrast, others have shown it to inhibit the enzyme. We therefore decided to further investigate this effect by caffeine on saliva alpha amylase activity and also on isolated pancreatic alpha amylase activity by using a spectrophotometric method. In order to determine the saliva alpha amylase activity, saliva samples were collected from 5 young people and added to test tube containing pH 7 trace buffer. After that, 1 milliliter of this solution was exposed to caffeine in concentration between 0 to 2% solution. 10 milligram per milliliter starch solution was added to the tube and incubated at 37 degrees for 30 minutes. The amount of produced by action of saliva alpha amylase was determined spectrophotometrically by using some reagent in 540 nanometer. Each experiment was performed in triplicate tubes on 3 separate occasions. Results are presented as means plus minus standard deviation. The significance of the differences between the various treatments and the control were analyzed using students' t-test. A probability level of p less than 0.005 was considered to be statistically significant. And in a very similar way, the activity of isolated pancreatic alpha amylase activity was determined. Isolated pancreatic alpha amylase enzyme was exposed directly to several concentrations of caffeine. The concentration were between 0 to 0.2% solution in a test tube containing starch solution with concentration between 0 to 20 milligram per milliliter and, of course, pH 7 trace buffer. And then the tubes were then incubated in 37 degrees for 30 minutes. The amount of starch digestion products by alpha amylase with and without the presence of caffeine was determined spectrophotometrically by using, again, some reagent in 540 nanometer. Each experiment was performed in triplicate tubes on three separate occasions. Results are presented as mean plus minus standard deviation. And again, the significance of the difference between the various treatment and the control was analyzed using students' t-test. A probability level of p less than 0.05 was considered to be statistically significant. And now we come to the results. Here in the first slide of the results, we can see this slide shows the results for saliva amylase activity. We can see that 1% and 2% caffeine solution reduced saliva amylase activity by 17 here and 30% respectively versus control. These results suggest that caffeine inhibits saliva alpha amylase in a dose-dependent manner. And to the second slide of the results. Here we can see the result for isolated pancreatic amylase activity. It's showing that 0.1% caffeine solution reduced isolated pancreatic alpha amylase activity by 27% and 53%. Here we can see it here and here, with 3 milligram per milliliter and 4 milligram per milliliter starch solution respectively versus control. So the inhibition level is dependent on the substrate concentration. And if we refer to this result shows the result for obtained with higher concentration of the starch of the substrate. You can see in the dose response curve that you can see indicates that 0.1 caffeine solution inhibited alpha amylase activity by approximately 30%, we can see it here, and 50% for the 6 milligram per milliliter and 20 milligram per milliliter starch solution, respectively, versus control. The 0.1%, it shows with the orange line here. And if we continue to discuss in the same dose response, 0.2% caffeine solution inhibited alpha amylase activity to a greater extent. We can see it here. Approximately 50% for the 6 milligram per milliliter starch solution, and 75% for the 20 milligram per milliliter starch solution. These results suggest that caffeine inhibition effect is dependent on both caffeine level and starch concentration. And if we look in this slide, we can see that 0.05% caffeine solution inhibited alpha amylase in several concentrations of starch, as was in the other slide. But however, with regard to the highest starch concentration, which is 20 milligram per milliliter of starch concentration, caffeine appears to have no effect on amylase activity. These results suggest that caffeine inhibits alpha amylase in a competitive manner. Well, what is the possible significance of these results? First, it was found that caffeine significantly reduced alpha amylase activity in a dose-dependent manner, both in saliva alpha amylase model and in isolated pancreatic alpha amylase model. Our results also suggest that caffeine inhibits alpha amylase in a competitive manner. We demonstrate caffeine as a food ingredient, with a potential to inhibit carbohydrate breakdown. Therefore, this finding may indicate a positive health benefit of caffeine. These findings are consistent with other studies suggest that consuming coffee regularly could be associated with a decreased risk of developing type 2 diabetes over the long term. Digestive system function suppression is known as one of the sympathetic nervous system effects on human physiology. Therefore, the inhibiting effect of caffeine on starch decomposition shown in this study is consistent with the effects of sympathetic stimulation of the digestive system, which are also known to be caused by caffeine. Thank you very much for your attention.